When we consider these enzymes, their activity can be controlled. Their activity can be controlled by competitors. By inhibitors, by inhibitors. These inhibitors occur in two kinds. We have competitive inhibitors that allows us to achieve competitive inhibition, and non-competitive inhibitors that allows us to achieve non-competitive inhibition, right? Now, let's consider an enzyme. Say enzyme 1. That enzyme 1 is part of a metabolic pathway, right? So that metabolic pathway involves the first substrate here. That first substrate is going to be converted to a product that is going to be the substrate for another enzyme, enzyme 2. That substrate is now going to be converted to another product right here. Product that is going to be the substrate for enzymes 3, and so on and so forth until we have the end product, right? Remember the case of the synthesis of an amino acid, right? It's not one single reaction, but multiple reactions. So now, when the cell has now so much production of the product, then we have a lot of the product here. That bunch of product can attach to one of the enzymes in the metabolic pathway. Most of the time, it's going to be the first enzyme or the second enzyme. In this very case, we are going to take the case of the first enzyme. Now, remember that the substrate is going to have an active site where it attaches, right? But some enzymes are going to have another site. That site matches a binding site for the product. When the product attaches to the other site, but the active site, what happens is that binding is going to cause an overall conformational change of the enzyme 1 so that the active site, the shape of it is going to change, and the substrate is not going to be able to recognize the active site anymore, or rather the active site will not recognize anymore the substrate. And now we have an inhibition that is caused not because the active site is busy, but because the active site is misshaped. This is what we call non-competitive inhibition. So the inhibitor is binding on another site but the active site, and that binding is causing a change in the conformation, which prevents now the substrate to bind on the active site. Again, non-competitive inhibition. That non-competitive inhibition is very common in the cell in the form of feedback inhibition. In the case of a feedback inhibition, the end product of a metabolic pathway is used to shut down the first enzyme or the second enzyme in the pathway that produces that very product, right? That's the feedback inhibition. While in the case of competitive inhibition, we typically use competitive inhibition to actually design drugs. When we design these drugs, we design this drug to look like substrate, but they are not the substrate. For instance, when we fight infections, right? We prevent, for instance, the synthesis of the cell wall of a bacterium. In a case of staph infection that we want to fight, and we provide an antibiotic, right? That is shutting down the production of staph cell wall. Basically, what we are doing is to design a drug that looks like the substrate that is part of making up the cell wall, and we use a lot of the drug compared to the number of molecules of the substrate, right? So we're going to do intentionally competitive inhibition by which the drug is going to bind on the active site in place of the substrate, right? That's competitive inhibition, because there is a competition for the active site. Okay, so know that feedback inhibition is the most common process by which the cell regulates the activities of its own enzymes, right? So that these enzymes are not doing work for nothing, right? When we don't need actually these enzymes to be doing work. Okay, so now the chemical reactions that happen in our cells need energy. That energy for a given chemical reaction is going to be different from another chemical reaction. And this energy that is needed for this chemical reaction function like price tags in a corporation, right? This price tag in a corporation are not set by the cashier. No, they are set somewhere in a corporation, right? Every single chemical reaction in our system has a level of energy that is needed for that chemical reaction to occur. Unless we have the amount of energy that is needed for that chemical reaction to occur, that chemical reaction will never occur, right? That energy is called activation energy, okay? That activation energy is the energy that is needed for that chemical reaction to occur, right? Again, in terms of comparison, it's like a price tag, right? Say you go to Kohl's and you say you want an Apple Watch, and that Apple Watch, it's say $399, okay, and 99 cents. And in your pocket, you have $250. That's what you have in your pocket, okay? So you go there at Kohl's with, you grab the watch and you go to the cashier. Will the cashier give you the watch? The cashier will not give you the watch because you don't have $299 and, $399.99. Say you decide to save some money. You save some money and now you have been able to save up to $397. Okay, that's what you have now in your pocket. You go to Kohl's and you ask for the Apple Watch. They tell you, well, sorry, you don't have $399.99, which is the price tag for this watch, so we cannot sell you the watch, okay? And you save some more money, and now this time you have, you have $500. So with your $500, you go to Kohl's, you go to the cashier, now they can sell you the watch. You can get the watch, right? Because you have that amount of money, right? $399.99, it's a bunch of money, right? And since you are a student, you are going to say, well, it's only reasonable to not buy a watch that is $399.99. So I'm going to wait for sales, right? So you wait for Memorial Day sales, right? And you are told that during Memorial Day sales, well, the watch that was $399.99, now it's going to be $210. Okay? That's the sale price. Okay? So you are going to do whatever it takes to go there at Kohl's during Memorial Day to actually get your watch that you could not afford normally, right? So enzymes in the cell are acting like this discount agents, right? They are decreasing the amount of activation energy that is needed for a chemical reaction to occur, right? So with enzymes, you only need a small amount of energy, right? Activation energy, which actually causes enzymes to be very powerful in our system. In fact, in presence of enzymes, the reactions are not only going to happen, they're going to happen faster and more often. Why? Because they lower activation energy. So here is one thing I want you to remember. Enzymes speed up chemical reaction in the cell by lowering activation energy. Let me repeat that. Enzymes speed up chemicals reactions in the cell by lowering the activation energy required for that chemical reaction to occur, right? Another thing I want you to remember is that most of the chemical reactions that are catalyzed by enzymes are intended to happen in the cell, with or without the enzymes. The question is, at which pace are these chemicals reactions going to happen? And what sets the pace? What sets the pace is the amount of energy that is needed for this chemicals reaction to happen and the amount of energy that is present in the cell, right? So you understand that a chemical reaction without enzyme can take forever to happen, right? Which, by the way, when you have a high fever and you have denaturation of your enzymes, you can die because this reaction takes that long, right? They take that long, so you have the time to die before this reaction can occur. That's why we cannot live without enzymes. Conditions such as acidosis are bad news for us, right? Very high fever, so hyperthermia, it's a bad news for us because our enzymes are losing their shape, they are being denatured, and they are not going to be able to do their job, so that these chemicals reactions are just going to take forever to occur. So much forever that you are going to die. Okay? Now, with that being said, remember the concept of activation energy and the role of enzymes. Now, at this point, let's take a look at the transport of chemicals across the plasma membrane. Remember that the plasma membrane plays many roles in our cells, including setting the limit between the inside and the outside of the cell, recognizing cells, each other, right? Being able to recognize each other because of their membrane and what they have on the surface of their membrane, right? Transporting chemicals in and out, right? Serving as receptors, right? Some proteins that are embedded in the plasma membrane are going to play the role of enzymes, right? And attachment, cell-to-cell attachment, to name only all these functions. Now, remember, plasma membranes are semi-permeable, right? Which means that some substances can cross freely the plasma membrane, while others cannot. They have to be transported, right? Now, some actually are so small, they are non-charged, they are non-polar. An example of those are, for instance, nitrogen gas, carbon dioxide, oxygen, to name only these three gases. These molecules are small enough to be able to diffuse across the plasma membrane freely. But most chemicals that we come across with, including ions, proteins, sugars, lipids, what else? Nucleic acids, they cannot cross the plasma membrane because they are simply huge. Okay? They are huge. Some of them are polar, some of them are non-polar, some of them are polar, some of them are, shaped in a way that do not allow them to get through. So, how do the chemical get through the cell, get through the plasma membrane. Three ways. One is the passive transport. The passive transport, overall, the chemicals are going to move from compartment of high concentration toward a compartment of low concentration. And the net movement is going to stop when you have equal concentration across the membrane. Two is that in that passive transport, energy is not required for that motion of the chemicals to occur. Energy is not required, right? I want you to remember that. The second type of movement is going to be the active transport. In the case of the active transport, first and foremost, you want to know that active transport costs energy. There is no active transport without energy. Remember that. Two, the second thing I want you to remember is that active transport requires a transport protein embedded in the plasma membrane. Let me repeat that. An active transport requires a transport protein to be embedded in the plasma membrane. Finally, in an active transport, the chemicals can be carried against their gradient. In other words, they can move rather than going from high concentration to low concentration. The cell can actually move the chemicals from low concentration to high concentration, which is thermodynamically challenging, right? And that's the power of using energy. When you use energy, then it's actually giving you the possibility to actually carry molecules against their gradient of concentration. And then we have the bulk transport. In the bulk transport, you have that the chemicals are going to be moved as bulk. And even huge particles can be moved. So here we have the cargo that needs to be enclosed in some kind of vesicle, and that vesicle is what is going to move the whole cargo either inside of the cell or outside of the cell. Now, let me tell you this. Bulk transport, it's a form of active transport, okay? Which means that bulk transport requires expense of energy. Okay, bulk transport requires expense of energy. We are going to address that. Now, let's take a look at, first of all, passive transport. In the passive transport, you have, first and foremost, simple diffusion. In simple diffusion, the chemicals are simply crossing the plasma membrane without any kind of support, right? They simply follow the gradient from high concentration toward low concentration. This is exactly what happens when you put, for instance, a piece of sugar in the water. When you put a piece of sugar in the water, okay, in this very case, a crystal dye, that crystal dye will start dissolving in the water and diffuse from the area of high concentration toward the area of low concentration, and eventually that diffusion is going to go on until you reach a certain level of equilibrium where you have an equal concentration of the solute everywhere in the solution, right? Now, remember, simple diffusion does not require energy. Examples of chemicals that can do simple diffusion are oxygen gas, carbon dioxide gas, and nitrogen, to name only these three. So now, besides simple diffusion, you have another passive process. That passive process is facilitated diffusion. That facilitated diffusion, it's a type of diffusion that is not simple, right? It is not simple because it is facilitated by a protein that is embedded in the plasma membrane, okay? That protein that is embedded in the plasma membrane has to be a carrier protein or a transport protein. That transport protein does not require energy to be activated. No, no, no. It's almost like a channel that is just open. That channel that is open is only going to allow chemicals that match the shape of the channel. If the chemical does not match the shape of the channel, that chemical will not go through. It's also going to discriminate based on the size of the chemical, right? So you have the size, the shape, and the charge of the chemicals that are going to be the conditions for the chemical to go across, right? So if you consider this solute that is out here, and this, by the way, a solute is the chemical that is dissolved in a solvent, right? And in our situation, the solvent of life being water, right? So this solute is here in the water and not at all present in the inside. Now, because the solute matches the transport protein here, so you see there is a match. That matching is going to cause a conformational change. That conformational change is going to be such that the transporter is going to release the chemical inside. As it releases the chemical inside, that net movement is going to stop whenever the concentration of the solute is equal on both sides of the compartment. Now, it is a fact that in our water-based environment where our cells find themselves in, a lot of the molecules are either charged, polar, or large. As a consequence of that, these molecules are not necessarily, these chemicals, I should say, are not necessarily able to cross the plasma membrane. Now, what happens is that most of the time, water is the one that has to cross the plasma membrane to compensate for the fact that this solute cannot cross the plasma membrane. In that case, the movement of water across the plasma membrane, while it is diffusion, we don't call it diffusion, we call it osmosis, right? So at this point, we are going to address the movement of water molecule across the plasma membrane. So osmosis, again, is the movement of water across the plasma membrane because the solute cannot cross the plasma membrane. So here, we are always going to ask the following question. What is the concentration of the solute? A given solute, it can be salt, it can be sugar, it can be amino acid, it can be any other chemical. What is the concentration of that solute inside of the cell versus outside of the cell? So we are going to consider that the cell is in some kind of bucket, right? That bucket being our body, or the body of the deer, or the body of an oak tree, right? And then we are going to ask, what is the concentration of the solutes inside of the cell versus outside of the cell? So because the concentration of the solute is more inside of the cell, what do we see? We see a net movement of the water molecule inside, right? And as these water molecules are going inside, what happens is that the volume of the solution inside is going to increase, right? And eventually is going to, the movement of water molecule is going to stop when we have an equilibrium, right? In the concentration of the solute, okay? So again, osmosis is the movement, the diffusion of molecules of water across the plasma membrane. Now, in real life, what does this look like? Well, what it looks like is that a cell can find itself in what we call an isotonic environment. An isotonic environment means that the cell being in a bucket, you have as much solute outside of the cell as you do inside of the cell. By the same token, here, that's for the animal cell. For the plant cell, same thing. You have as much solute outside of the cell as you do have inside of the cell. So this is the ideal condition metabolically for the cells to thrive. The cells want to be in an iso means equal tonic concentration, right? In the solution, all right? So now, the cell is not always in an isotonic environment because guess what? You always have salt, you have amino acids, you have sugars that are always either inside of the cell or outside of the cell or both, right? But we do know for sure that these chemicals cannot freely cross the plasma membrane. Water molecules are the ones that have to cross the plasma membrane. So now let's look at the consequences of the difference in the concentration. So if our cells find themselves in a hypo means less, tonic, concentrated, right? If our cells find themselves in a less concentrated environment compared to the inside, for red blood cells, water is going to rush inside, okay? When water rushes inside, there is a point in which, because the red blood cells do not have a cell wall, these cells are going to burst, okay? Which is a bad news, right? You understand that. But interestingly, for plant cells, the plant cells are going to take up volume, but these cells are not going to burst. Rather, these cells are going to be very inflated. That inflation of the cell is going to cause plant cells to be stiff, right? Upright, well, a very healthy plant, right? This is actually the ideal situation for most plants, right? That's why we have here that the cell is in the normal turgid cell position, right? Now, this is in a hypotonic environment. What if the cell was in a hypertonic environment, such as, for instance, the marine environment that has a bunch of salt, right? You know that plants are found in aquatic environment and animals are found in aquatic environment, right? What is the result in the cells? Well, here is the result. In a right, tonic concentrated, in a more concentrated solution, hypertonic environment, the water is going to leave the cell to go outside of the cell so that the cells are going to shrink in size. They're going to shrivel, right? Now, how about plant cells? Plant cells are going to shrink, but overall, the plant shape will not change. Why is that possible? Because of the cell wall, okay? However, in terms of how the plant is going to look like in these conditions, the plants are going to be like this. You know, rather than being like this, they're going to be like this, okay? This is what happens during hot and dry days, right? During hot and dry days, the plants lose water so that the outside environment becomes hypertonic. Water is going to leave the inside of the cell to go outside of the cell, and overall the plants are going to be like this, okay? All right. So how about active transport? Active transport, again, requires input of energy. And most of the time, if not all the time, that input of energy is going to be in the form of ATP, right? Now, that's one. Two, these active transport systems are going to require transport proteins. And these transport proteins are going to carry substances based on the shape of the transporter. Okay? Which means that these transporters are going to be specific, right? One that is able to transport amino acids will not know how to transport nucleotides, and so on, right? And finally, this mode of transport can carry molecules against their gradient, right? When ATP is spent here, okay? ATP is spent here. Notice that the expense of ATP is going to cause a change in the conformation of the transporter, which actually matches now the ability of the solute to attach to the transporter. And now, when ATP is released from the transporter, you have another conformational change, which releases the chemical inside, right? A lot of our ions, such as potassium and sodium, are transported in and out of the cell that way, okay? Sodium potassium pump actually work that way, carry molecules against their concentration, or rather carry substances against their concentration. Finally, but not least, let's address bulk transport. The first stop we are going to make is going to be exocytosis. Exocytosis is to take chemicals from the inside of the cell toward the outside of the cell. For instance, when we consider the synthesis of antibodies, after the antibodies are synthesized, they are sent outside of the cell in circulation so that they can go after antigens, right? By the same token, when sugars are synthesized, the sugar that make up the extracellular matrix, right? When they are synthesized inside of the cell, they're going to be sent outside of the cell so that they can go play the functions they were designed to play, right? So you have transport vesicle like this, which are basically phospholipid bilayer with cargo inside, right? So this cargo are going to be sent out by way of the phospholipid bilayer of the vesicle coming and merging with the phospholipid bilayer of the plasma membrane, right? Eventually, the cargo are going to be released outside of the cell, okay? That process requires expense of energy in the form of ATP. Then we have endocytosis. Endocytosis is the opposite of exocytosis. In endocytosis, the cell is going to bring some particles in, right? By forming an invagination that you can see here. You see the cargo molecules, okay, that needs to come in. You have a vesicle that formed. That vesicle buds off, right? And then you have a transport vesicle that comes in here. Now, endocytosis occurs in three forms. We are going to have phagocytosis, which is also called cellular eating, okay? Cellular eating, okay? That cellular eating consists of surrounding particles, engulfing them, and digesting them. This is how, for instance, our white blood cells are able to engulf bacteria or viruses, break down those viruses and bacteria, and that's how we get rid of the infection, right? By phagocytosis or cellular eating. The second type of endocytosis is pinocytosis. In the case of pinocytosis, we're going to have the vesicles that are going to form around liquid substances, okay? And these liquid substances are going to be engulfed and be brought in. This is also what we call cellular drinking. Cellular drinking. Finally, we do have receptor-mediated endocytosis. In receptor-mediated endocytosis, on the surface of our cells, we have receptors. On these receptors, the cargo molecule is going to come and bind. For instance, how do we internalize our cholesterol? Well, we internalize cholesterol because cholesterol combine with some proteins forming a structure that we call kilomicrons, right? These kilomicrons are recognized by receptors. Upon binding of the kilomicrons on receptors, you have a number of receptors that receive the kilomicron, and there is a certain threshold that when reached, you have an invagination that is going to happen so that the whole structure is going to be engulfed and brought inside, right? This is how, this happens. So we have that vesicle that forms that vesicle is going to have some proteins attached to that vesicle and allow that vesicle in that way. So you can see here the cargo molecules that are attached to receptors, and you have the coat here. That's a coat of clathrin. Let me spell that. Clathrin. Okay. Well, did I spell it right? It's either like this or like this. Clathrin, but I cannot remember exactly the correct spelling. It's one of the two, okay? And sorry, I'm going to verify. So with that being said, you have the receptor right here. You have the cargo molecule here. And overall, this is going to end up forming a vesicle, and that vesicle is going to bud off and migrate inside. And at that point, the fate of the cargo is going to be defined by, the need or the nature of the very chemical that was brought inside. Now we're going to conclude this chapter with reminding you to do a synthesis of a summary of this chapter, right? Do a summary and test your understanding of this chapter by doing questions at the end of the chapter, by designing also your own questions, by talking with other students in this class. Say you have designed some questions, you have tested your knowledge based on the question you designed. You also contacted a classmate who also designed their own question. Do some kind of exchange of questions. That way, you can test if you can actually answer questions that another student designed. Finally, but not least, I highly encourage you to design handwritten concept map that is going to help you connect the dots and understand all of this. All right, so thank you for your attention. Talk to you in the next chapter. Bye now.